Hello, I'm Leo Choi, a scientific researcher from Glyceum Inc., and I'll be presenting you today on alleviation of obesity via enhancing mitochondrial function by HSG4112. Here's a disclosure of conflict of interest. I'll first describe HSG4112, which is a small oral molecule discovered through in vivo phenotypic screening, and describe its efficacy in high-fat diet-induced obese mouse model, and the transcriptome analysis we performed to investigate the drug's potential mode of action. Here, we acquired the clue that the molecule may be involved with mitochondrial function, so we performed a series of in vitro mitochondrial function analysis and mitophagy assessment. Ultimately, the conclusion of this presentation is the establishment of the mode of action of HSG4112, which is mitochondrial function enhancement for the treatment of obesity. HSG4112 is based on glabridin, the main chemical component of licorice. Despite reported beneficial effects to the metabolic syndrome, glabridin has not been developed as a drug due to its intrinsic limitations as a molecule and lack of established synthesis method. Glyceum Inc. developed chemical synthesis method and performed an SAR study to optimize stability and efficacy. The library of 300 derivatives were tested directly in vivo mouse models of obesity to observe direct effect on weight loss, as shown in this example graph. HSG4112, which showed vastly different chemical profile and about threefold greater efficacy compared to glabridin, was selected and is currently in phase 1 clinical trial in South Korea. To examine the drug's efficacy, a 6-week study was performed on high-fat diet-induced obese mouse. All groups were fed high-fat diet except the normal control group shown in gray here. For positive control, we used semaglutide, which is a prominent GLP-1 receptor agonist in the field of anti-obesity drugs, at its maximum efficacy dose. In terms of body weight, compared to the vehicle-only group shown in red here, HSG4112 in light and dark blue dose-dependently reduced body weight up to full normalization at 100 mg per kilogram, which was superior to semaglutide shown in orange. The body composition measured by NMR showed significant reduction of about 80% fat mass and full retention of relative lean mass. Food intake was significantly reduced in all groups at a similar level, while the energy expenditure measured by the indirect calorimetry was also increased in all groups at a level equivalent to the body weight loss, suggesting that energy expenditure must have played an essential role in causing the difference of weight reduction. Next, we performed transcriptome analysis on terminal muscle, liver, and white adipose tissue to gauge any clues as to the drug's potential mode of action. So we focused on upregulated genes as changes mediated by the drug, and ranked the most significantly enriched gene ontology terms as shown here. In muscle, most of the 1,411 significantly upregulated genes were located in mitochondria and its membrane, and mediated the biological process of protein metabolism and catabolism, and also mitochondrial respiratory chain complex assembly. The same was true in the liver, where most upregulated genes were localized to mitochondria and its inner membrane, and mediated the biological process of amino acid metabolism and catabolism. In the white adipose tissue, the 3,355 upregulated genes were mainly affected by the loss of weight, because all groups that had weight loss compared to the vehicle group showed similar patterns of highest ranked gene ontology terms. So we ended up specifically drawing a heat map of genes that form the mitochondrial oxidase complex in the white adipose tissue. And we found that compared to the vehicle group's profile on the leftmost column showing low expression level, HSG4112 in the third and fourth column normalized and generally increased the expression level similar to that of the normal control group. But semaglutide on the second column showed similar expression level to that of the vehicle group in spite of the observed weight loss effect. Overall, we acquired the clue that the HSG4112 may be involved with mitochondria and its function, so we dived in deeper by doing in vitro assays in SHSY5Y human neuroblastoma cells, which are commonly used for this purpose. And MPP Plus was used as mitochondrial dysfunction inducer. We first performed cell-based mitochondrial activity profiling system assay, where we assess the NADH dehydrogenase activity, the intracellular ATP content, the cellular reactive oxygen species or ROS level, and the mitochondrial ROS level. And we found that HSG4112 compared to the negative control remarkably increases NADH dehydrogenase activity and ATP content, all the while reducing cellular and mitochondrial ROS level. Also, we measured the rate of oxygen consumption, which is the prime action of mitochondrial oxidative phosphorylation using Seahorse Analyzer. While MPP Plus significantly reduced the overall OCR profile, HSG4112 dose-dependently increased the basal respiration rate, the ATP turnover rate, and the maximum respiratory capacity, restoring the damaged OCR profile. So, because we observed dose-dependent improvement in all parameters, we found that HSG4112 increases and improves mitochondrial function. Lastly, we investigated the drug's effect on mitophagy, which is mitochondria-selective autophagy as a quality control system for mitochondria. We saw that HSG4112 dose-dependently induced mitochondrial fragmentation shown in red here, and increased autophagism formation shown in green here, which merged together to suggest the presence of mitophagy, all the while maintaining cell viability. This was distinct from the positive control CCCP, which is a mitochondrial uncoupler which induces mitophagy and also cell death. We further tested whether this is mitochondria-specific autophagy by using ATG5 knockdown cell for ablation of autophagy and DRP1 knockdown cell for ablation of mitochondria-specific autophagy. And in the end, we were able to verify that HSG4112 induces mitochondria-specific autophagy or mitophagy. In conclusion, we were able to establish the mode of action of HSG4112 as mitochondrial function enhancement. Mitochondria modulate both energy production and cellular inflammation, both of which are critical factors in treating metabolic diseases. But so far, no drug has been successful at targeting mitochondria for the treatment of obesity. We believe that creating healthy mitochondria ultimately increases energy expenditure and decreases inflammation for the treatment of obesity. But in this presentation, we show a portion of this story where HSG4112 enhances mitochondrial function as its main mode of action, increasing ATP production, oxygen consumption, and mitophagy while decreasing mitochondrial and cellular ROS level. We were able to recently discover the target protein of HSG4112, which is not disclosed here. Our future directions are to confirm this mitochondrial function enhancement in different cell types and to further establish the full molecular mechanism of HSG4112. Thank you.